Hey, it's Taki, and welcome to RDU Airport. Before we go in depth about their immigration status, we must first have an understanding of epistasis. Epistasis is a type of gene interaction where two or more different genes on a different loci contribute to the same phenotype in an unexpected manner. Each gene has a specific product that determines the phenotype of that specific trait, which can be altered as a result of masking, etc. Capital A in our example is a dominant epistatic gene. In the presence of capital A, masks or alters the expression of gene B. However, when it's recessive AA, it will not have an epistatic effect over the phenotypic expression of gene B.
Now, before we look at the passport, we must look at the protocol. You have the passport. Once you have the passport, you have the first gene. Little a will mean that you can pass through to TSA. Big A, anything, means that you are stuck at the airplane. Now, once you pass through TSA, you have the second gene. Here, if you have a dominant B allele and anything else with it, you are more than welcome to enter the country. If you have both recessive, you are stuck at the airport forever. If you enter the country, you cannot leave and you cannot go back.
Hi, so right here I have the passport. This passport contains the genetic makeup of this duckling. Within the passport, we have pages. These individual pages resemble genes, and within those genes, you have the alleles. These alleles will help determine the immigration status of this duckling.
All right, here we have the passport for this duckling. Once we open the passport, we see that for gene number 1, we have an allele of little a, which means they can now pass and go through TSA. We turn the page for the duckling and we find that on gene number 2, they have an allele of big B, which means they can pass and enter our country. Now we have this puppy. We open the passport for this puppy and we realize that on the first gene, we have a big A, which is an inhibitor, which means this puppy cannot get off the airplane. When we look at gene 1, we have an allele of little a, which means this Dumbo can move on to TSA. We turn the page and we look at gene number 2, we have an allele of little b, which means Dumbo is going to be stuck at the airport forever. So here's the dihybrid cross. As you can see, dominant A, dominant B will be labeled in circles. Dominant A, 2 recessive Bs will be labeled as triangles. And recessive A and a dominant B will be labeled as stars. And then recessive AA and recessive BB will be labeled in a square. The circle and the triangle will both be stuck at the airplane because of the fact that they both have a dominant A, which means they won't pass the first checkpoint. Now, this one has a little a, which passes the first checkpoint, and then it has a dominant B, which means they will now enter the country, which means these 3 people are welcome to enter. Now here you have a recessive AA and recessive BB, which means they will pass the first checkpoint, but once they get to TSA, they can't enter the country because of the recessive BB, which means one person will be stuck at the airport. The ratio is 12:3:1, which is single dominant epistasis, and normal dihybrid ratio is 9:3:3:1. This is the other cross. As you can see, circles are big A, big B, which is 8. Stars are little a, big B, which is 8. Little a, big B makes it all the way through. Big A, big B does not make it off the airplane.